So welcome to the Natural Areas Association 2021 webinar series. This is the last of the year. We're excited that we were able to offer 10 wonderful webinars, and we look forward to scheduling our 2022 series. So we hope you'll continue to join us going forward. I'm Maura McGinty-Kloss, and I'm the NAA's Program and Outreach Manager, and I'm delighted to be joining you today. So the purpose of NAA webinars is not just to convey information, though there's a lot of great science here. The purpose is also to help you engage with your peers and colleagues so that we can talk about the topics that are important to you. So I hope that you'll engage in chat and share best practices, share things that you know, and create a two-way dialogue during this webinar. If you do have questions or thoughts or things you want to share, please post them in chat. I will be monitoring chat throughout the program, and I will also address as many questions with our speaker as possible at the end. And if we don't get to all of them, I will be sharing her contact information. So if there's things that you'd like to follow up on, you can. So I just want to tell you a little bit about the Natural Areas Association. We are dedicated to the management and restoration of ecologically significant landscapes in North America, because we understand that protecting nature requires reliable science to inform your practices and on-the-ground practices, and a network of stewards who tirelessly work to manage and restore land and biodiversity. We're a community of peers who become a resource to each other, and we encourage you to use each other to ask questions, propose ideas, share methodologies, and advance best practices. NAA supports you with a variety of things, such as these webinars, but also with things like our Natural Areas Journal that's a peer-reviewed journal that comes out quarterly, our Stewardship and Action Field Workshops, and our State Natural Areas Programs Roundtables, along with other events that we might host throughout the year. If you are a member and you are participating in this webinar, you know that all webinars are no cost to our members. You will have access to an archive of this recording and all of our webinars, Natural Areas Journal articles, and other resources. If you paid to participate today, I just want you to know that you will automatically receive a 7-day trial. And if during that trial you elect to join us, a year's membership is $79. We will apply the $29 to the $79 annual membership, and you'll be able to join for $50. And not only when you get to see this on-demand recording, but you'll have access to all of our on-demand recordings and everything that's available to members. I want to take a moment to talk about our in-person Natural Areas Conference. We're so excited. It's been 2 years since we've had an event. And we are thrilled that Robin Wall-Kimmerer will be one of our keynote speakers. And it's going to be September 6th through 9 in Duluth, Minnesota, which is a gorgeous part of the country. Also, Duluth is located right on the shore of Lake Superior, of one of the Great Lakes, and is renowned for some amazing natural areas. So we hope that you'll consider joining us. The conference is September 6th through 9, which is the week right after Labor Day weekend, so it's perfect to build in a little vacation time. We also just want to remind you all that the call for proposals is now open. The deadline for a proposal is March 1st, but we hope you won't wait that long because we are excited to see what you all have to share. So please, we're still looking for breakout sessions, hands-on workshops, posters, all of those things, and we've made the application short and sweet. So I will post in chat after when our speaker gets started a link to the call for proposals, and we hope you'll consider submitting one. I also want to talk to you a little bit about our February webinar, which is What's That Smell?, which is how detection dogs can lend a paw in conservation programs. And I want to mention that you may have noticed I didn't show a January webinar, but we will be hosting, you're hearing it here first, a Stewardship and Action virtual field workshop called Sustainable Summits, Managing Public Access for Protection of Rare Plant Communities. It is a field workshop that we actually conducted in person in September, and created video of all of the presentations and all of the work we did outside, and we're putting it together in a virtual presentation in Zoom that will be hosted on January 25. So we hope that you'll note that on your calendar and watch for NAA emails, and we'll be sharing more information. And now let's get started with the program that we have planned today, which we're very excited to welcome our speaker today, who's going to talk about rare plant propagation and reintroduction, questions and considerations for natural and historic resources lands in New Jersey. So Elizabeth Olson works for the New Jersey Department of Environmental Protection and the Office of Natural Lands Management, where she assists in the field surveys for rare plants and researches the ecology and management of state-listed rare plant species. Liz is also an adjunct instructor at Stockton University, and she studied ecology and environmental sciences, earning a BS from Temple University and an MS from the University of Maine. So now I am going to invite Liz to take over, and we look forward to your presentation today.
Great, thank you so much, Moira. Hello, everyone. Thank you so much for the welcome. Thank you, Moira and the Natural Areas Association for hosting this webinar. So Moira here is, before I get started, Moira is going to put up a poll for y'all to answer, just to get a sense of your experience with rare plant reintroductions. So I'm here today to talk about this research report that I wrote for my employer, the New Jersey Department of Environmental Protection. This is a report that provides a detailed overview of the benefits, risks, and challenges of propagating and reintroducing rare plant species. And it summarizes some of the major concerns and considerations that have been identified in the current scientific literature. And the PDF of this report is available online at this web address, but Moira said that she would be posting this link for you all to access later, and a couple other resources on the topic that you might be interested in. So don't worry about jotting that down. But right up front here, I did want to stress that the message here is going to boil down to, yes, rare plant reintroductions, they're tricky, they're challenging, they often fail, but they are sometimes very appropriate. And this document can provide some resources to help determine if that's the case. And then questions and topics to help you think about how to proceed carefully with planning a reintroduction project. But before I get too deep into it, I did want to quickly go over some of the terminology that I'll use. So within the field of plant restoration ecology, there are a lot of terms used to refer to the practice of outplanting propagules of rare species to restore their population. So you'll see some projects refer to an introduction, an augmentation, a relocation, or a transplantation. And these are just a few of the terms that exist. And each one does have a specific meaning. But for the sake of simplicity, in this presentation and in the written report, I just use the term reintroduction as a broad blanket term to encompass all of these types of outplantings of rare plants in restoration projects. And this type of document, this overview of rare plant reintroductions, was identified as being needed in New Jersey because over the years, the Office of Natural Lands Management, which is essentially our state heritage group, they've been asked to give advice and input on various proposals to reintroduce rare plants, either on state land or using rare plant material that was collected on state land. So, these are the things that have been proposed. And many of these proposals are often from folks who are looking to help conserve rare plants. There's a passionate group of plant conservationists here in New Jersey from all walks of life, really, from professionals to enthusiasts. And so often the proposals come from this group of conservation-minded folks who are looking to help make a difference in rare plant conservation. But additionally, developers have also proposed to relocate rare plants as a way to quote unquote mitigate habitat destruction. And also as climate change progresses, We are preparing for the potential that future proposals might suggest this practice of assisted migration. And that would be where the rare plant species would be planted outside of its native range in the hopes that the future climate there would be better for them. But whatever the motivation is, they're always, attempting to propagate rare plants or transplant them and place them back into natural habitat. But it was hard to know how best to respond to these requests. So this research was conducted to have a better understanding of the science of rare plant reintroductions and to have a way to evaluate reintroduction proposals. So in my research for this report, I conducted a thorough literature search, and that included overview, papers, and meta-analyses, as well as case studies and experiments that focused on single species reintroduction attempts. Also, documents that were more about policy and guidelines for the practice, and websites from state and federal governments, as well as nonprofit organizations. So that brought me to a total of about 163 references. This was certainly not a completely exhaustive search on the topic, but it was still very thorough. So I want to just take a step back to put this issue into context, and I'm guessing that I'm speaking to the choir here, but we all know that plants are at a major risk of extinction. And this recent paper from 2020 estimated that worldwide 39% of plant species are in danger of becoming extinct in the wild. And at the population level, local extirpations are also a big concern because they're much more frequent and widespread than species extinctions. And there is growing evidence that local extirpations can degrade ecosystems and break down food webs. So they're, it can be just as important. And the main cause of species loss is habitat degradation. And that can include things like fragmentation, land use change and development, and invasive species, all things that lead to habitat degradation. And also, of course, climate change is a big reason why plants are threatened. So with all of these threats that plants face, the on-site or the in-situ conservation of rare species, it's just not always possible, especially in extreme cases of habitat destruction or land use change or land conversion. Altered and degraded habitats are so common now that traditional conservation measures are not always enough for populations to recover. So it's just going to take a wide range of approaches to slow and halt the process of losing our rare plant species. And for example, botanical gardens and seed banks have been instrumental to conserving rare plants. And another solution can potentially be the propagation and reintroduction of rare plant species. However, all practitioners of rare species management are in agreement that the in situ conservation of rare species and their habitats is the best insurance against losing those species in the wild. The Center for Plant Conservation has worked for decades on the science of rare plant conservation, and their scientists have implemented many reintroduction projects, but even they state that they do not support or promote reintroduction as an alternative to in situ ecosystem protection. And at the federal level, reintroduction is a part of some federally listed recovery plans. And the Endangered Species Act recognizes that habitat protection is crucial. So they do support reintroductions in some cases, but always, the basis and always recognizing the habitat protection is the most important thing. And so in all of the 100+ resources I read, they all agree that rare plant reintroduction should be a last resort option. So in order to conserve plants, our first priority has to be protecting habitat by addressing the threats that rare plants face and the factors that are responsible for their decline. And really, by protecting the whole habitat, that's going to result in saving many other species as well, in addition to that target species. But for some species that are on the brink of extirpation or extinction, a reintroduction project might really be the only option left. So reintroductions can be an important tool for the recovery of our rare, threatened, and endangered plants. The goal of reintroductions and reintroduction projects is to establish resilient, self-sustaining populations that contain the appropriate amount of genetic diversity that's needed to adapt to changing environmental conditions, including climate change. And in this way, ultimately, to prevent rare species from going globally extinct or being locally extirpated. And in recent decades, the fields of restoration ecology and reintroduction biology have advanced. So there are some wonderful rare plant reintroduction success stories. When they are successful, they can increase the species populations and restore the historical range of species that have been declining or extirpated from some locations. Across a landscape, reintroductions can decrease the fragmentation of populations and kind of restore the metapopulation dynamics. And this can work to promote genetic exchange among populations, which can improve the genetic health of these dwindling populations. And at the community level, reintroductions can increase biological diversity to a site and can help return plant communities to their former composition. However, although reintroduction projects do have the potential to do all of these great things if they work, many respected biologists have cautioned that there's also significant concern about potential negative consequences or implications of conducting reintroductions. So we don't want to think that we can rely on reintroductions. And firstly, because they're often not successful. But there's a danger in shifting the focus of conservation work away from the root cause of species declines, which is often habitat destruction. Rare species that are declining are often responding to a negative shift in their habitat, which could be addressed directly with the appropriate management. And reintroduction projects could give the false impression that intact natural habitat is unnecessary for the preservation of species. You know, people can wrongly assume that moving plants is easy and inexpensive, when in fact, reintroduction projects are actually incredibly complicated and often unsuccessful. And protecting plant populations in their natural habitats could come to be seen as an unnecessary hindrance to development. And reintroductions could then be used to rationalize the destruction of natural habitat. For example, in Western Australia, there was a state-funded research project, and they were able to successfully develop propagation techniques for a rare orchid. Unfortunately, that was then used to justify the destruction of one of this orchid's last few remaining populations. And this is an example that was cited in Falk and Allwell, 1992, although I couldn't track down, what was that specific orchid species. But this shows that the messaging around rare plant reintroductions should be that natural habitats are essential for the survival of rare plants. Reintroductions can be attempted as a last resort when the damage has already been done, but they should not be used as a way to rationalize destruction of natural habitat. And creating new populations can end up obscuring the natural range and the ecological patterns of plant species if they are unknown to other professionals. And this has actually occurred for at least 2 species that I found information about, and shown on the screen here is the rare Plymouth gentian and the rare hard shield fern. And in both of these cases, professionals were studying what they believed to be new occurrences of these species. But in both cases, the new occurrence turned out to be an undocumented outplanting, an undocumented reintroduction. So there was confusion and wasted time and effort, all due to a lack of communication, really, between or among the natural resource professionals who work in the region. And it is really important to know if we are looking at a natural population of a rare species or if it's a planting, because if introductions if they artificially and temporarily increase the number of populations, then the species could abruptly lose any legal protections that it might have had. A rare species could suddenly be downlisted based on created populations, but the long-term survival of a reintroduction is very uncertain. So all of this is to say, our messaging around rare plant reintroductions just needs to really recognize that it shouldn't be at the top of the list for restoration actions. Really, habitat conservation and management should be first priority, is absolutely necessary. Habitats are just not easy to replicate. And also, we probably shouldn't be engaging in these like solo missions to place rare plants out in wild habitats, without really consulting and communicating with our fellow professionals. So in addition to those potential negative implications, there's also concrete risks and challenges of reintroduction projects. The process of collecting rare plants and propagules, it can be harmful to that source population and the source site, because removing any plant parts can cause reductions in its abundance or reduce the genetic variability of that population, and possibly result in an increased risk of extinction or extirpation. Because basically any plant material that's collected and used in a reintroduction, there's sadly like a good chance that it won't survive to produce another generation. And damage to the outplanting site can also occur. There can be trampling, soil disturbance, and possible introduction of invasive plant seeds or plant pathogens that could be transferred to the new site during the outplanting process. And we have to consider the genetic structure of rare populations. There may be locally adapted genotypes that are critical to the species survival. But unfortunately, at each stage in the reintroduction process, there is the potential to lose genetic diversity or negatively affect the genetic structure of the rare plant population. And rare plant reintroductions are inherently challenging because not often, and usually not enough, is known about many rare plant species and their habitats. These unknowns can include things like what are their pollination systems? What are their germination requirements? Are there important mycorrhizal symbionts, seed dispersers, and what are the specific microhabitat needs of the species? And actually not understanding the specific microhabitat needs is one of the main reasons why reintroductions don't succeed. This figure here is from a meta-analysis of rare plant reintroduction projects, and it shows the reported reasons why different reintroduction projects failed. And a lot of the reasons boil down to lacking a full understanding of the habitat requirements or the species biology. All right, so other challenges include the fact that reintroduction projects are time consuming. They can take many years of just planning and preparation just to lay the groundwork for a well-designed project. And then the outplanted populations might take years or even decades to stabilize after their initial establishment. They'll often require long-term maintenance. And even with all of that effort, success is still not guaranteed. And that feeds into the last challenge faced, which is that reintroduction projects can be quite expensive and will typically require funding for each step in the process. And because of the long timeframes that are needed to implement and monitor results, there's bound to be some uncertainty about being able to maintain that long-term funding. So knowing all of these risks and challenges, there are plenty of cases where reintroductions are necessary to the preservation of a species. So first we need to ask if a reintroduction project is justified or not. And the Center for Plant Conservation has excellent guidelines for determining whether a proposed reintroduction should proceed or not. So this is straight from their 2019 publication. And they advise that a reintroduction is not justified if it will undermine the imperative to protect existing sites. So as I mentioned before, it's generally agreed upon that the priority needs to be habitat protection. So if a reintroduction is proposed as an excuse to not conserve and protect habitat, then it should not proceed. And if existing threats have not been minimized or managed, then reintroducing a rare plant is not recommended because it will still be under the threat of extinction or extirpation until those threats are managed. And likewise, if suitable habitat is not available or sufficiently understood, then it wouldn't be worth implementing a reintroduction. And if the source population can't sustain removal of individuals or propagules, then it's just not worth the risk of permanently damaging the source population. And finally, if the species has not been thoroughly researched, especially its conservation status, and having an understanding of if it's actually in decline or not, if that's not understood, then it just wouldn't be reasonable to begin a reintroduction program when it might not even be necessary. So in all of these cases, a reintroduction is not justified, should not proceed, not recommended. Okay, and so, reintroductions, this should be a last resort, but they certainly are situations where it makes sense to attempt to reintroduce a rare plant. So the Center for Plant Conservation advises that a reintroduction may be justified if all of the following conditions are met. So first, if the distribution of the species is known and the populations are all small and in decline. And if the in situ management options are not enough for the long-term conservation of the species. Also, if the threats to the species have been identified and there's still high risk of extinction or extirpation. So for example, the in situ conservation can't be guaranteed at a site where, for example, the effects of climate change are too severe, for instance, in the case of rising sea levels. So if all of these conditions are met, then a reintroduction may be justified. And you can proceed with caution. All right, so Hawaii has many examples of reintroduction projects that were justified. And there's sadly a lot of examples from Hawaii because they're just fighting against both high numbers of invasive species and high endemic species lost. So for example, Alani, shown here on the left, is an endemic species, and it was reduced to 1 wild individual and 1 in cultivation. And another example here shown on the right is Hawaiian prickly ash. For this species, there were only 3 remaining individuals, and they were all isolated from each other, and they weren't within the range of its native pollinators. So the recovery efforts for these 2 species are still in the early stages, but the research is reporting some early successes. All right, so if the reintroduction is justified, then there are many topics, many components of a project that need to be tackled in planning a reintroduction project. So components, the components of a good reintroduction project. Our report goes into detail about these various topics and components that need to be addressed. But I do want to stress here that it is not intended to be like a how-to guide for reintroductions. I want to reiterate that that's just not the purpose of this document, but we do provide plenty of other resources that deal more with that, how to go about a reintroduction. But here are some of the important elements of a well-designed reintroduction project. So first, before even beginning to plan a reintroduction, it's really important to have a thorough understanding of the species. And this includes its conservation status, such as both the historical and the current distribution, and why exactly is the species rare, and what are the threats it's facing, for example, habitat loss, or the legacy of fire suppression, or invasive species encroachment. And we need to know the details of the biology and ecology of the species, things like its seed production, dispersal mechanisms, germination requirements, etc. And understanding the species genetics is important too, since the primary goal for restored plant populations is for them to be adapted to local conditions and to be resilient to environmental change. So it can be important to know if the remnant species populations have lost some genetic diversity. So here I want to mention the case of the Tennessee purple coneflower. It's endemic to a very small region in central Tennessee. And the recovery and reintroduction research included some genetic testing. And they found that it has naturally low levels of genetic diversity. So from that, they knew that the seed can be mixed from multiple subpopulations without fear of genetic depression in reintroduced populations. So this map here shows where Tennessee coneflower occurs. You can see how small that area is in central Tennessee. But you can imagine, though, a different outcome for a rare species with a wider distribution and more disjunct subpopulations with no gene flow among them, like this, for example. So it's just important to have some understanding of the species genetic diversity, because a rare species with this type of scattered distribution would more likely have very locally adapted genotypes, and it could suffer from genetic depression if those seed sources were mixed for reintroduction. And finally, knowledge of how the species can be propagated is important. Some species are really difficult to propagate and can require a lot of work to break seed dormancy. And one example of that is shown here, the Ruth's golden aster. Its seeds have a complicated process to break dormancy. So researchers found that first the seeds have to be dried and then washed with alcohol for a minute, then passed through a flame, then washed with bleach for 20 minutes, and finally rinsed 3 times with fresh water before they're placed into their soil medium. So it's a very precise and involved process just to break seed dormancy. So then the topics that a proposal should address for implementing a rare plant reintroduction project. It has to start with a good experimental design. And the reason for this is that the results will be better able to pinpoint which methods led to success or failure and why that was the case. All projects need to find an optimal sites for outplanting, which isn't easy, but they really need to make sure they've identified suitable habitat because, as I said earlier, inappropriate habitat is the primary cause of reintroduction failures. And I wanted to share one interesting example that illustrates that. This species shown on the screen here is Harperella. It's a federally endangered species found in the southeastern US states. But reintroduction efforts in Maryland and West Virginia found that this species is especially picky about its habitat. So first, it needs at least 6 hours of full sunlight daily. And it needs to be on streamside cobble bars that never dry out. It also requires minor periodic flooding, and yet it has to be in microsites that are protected from severe flooding because it can tolerate brief flooding events, but only if the water is not too deep. And also only if the flooding doesn't happen during the time of year where this species is actively flowering or in seed development. So a very Goldilocks suite of conditions. You can imagine it was not easy to find, this precise set of conditions to outplant the species. And we can't overlook the species interactions that might occur between the rare plant, target species and the other organisms that they rely upon. That can include its pollinators, its seed dispersers, mycorrhizal fungi, among many others. And a good example of this is shown here. This species on the screen is the salt marsh bird's beak. And there was a successful reintroduction of this species in California. But it was dependent on the presence of its preferred pollinators, which happen to be solitary burrowing bees. And the species is also a hemiparasitic plant, so it has to be growing next to specific native perennial plant roots for it to parasitize. So the researchers were able to discover this. They used an experimental framework and they learned through adaptive management what those other species were that were necessary for the salt marsh bird's beak, successful reintroduction. And next, there are a lot of details with the outplanting process that have to be considered because the survival of the plants can depend on planning out details like where, when, and how often to plant. And also, should the project be using seeds or would seedlings be better? How old should those seedlings be? All these details are important to consider because they will influence the future health and success of the population. Okay, so long-term monitoring is essential to reintroduction projects. It's been shown over and over again that there may be successes in the early years, there might have survival in the first few years, but these absolutely don't guarantee long-term success. So researchers have to have a strategy and a plan for long-term monitoring. And it's through that regular monitoring that they'll be able to catch signs of decline before it's too late to correct for, like Often the plants might need to have supplemental watering, or they might need to be protected from herbivores until they fully establish. So going back to the example of salt marsh birdsbeak, this is actually a great example of the need for long-term monitoring. The researchers found that this species has these really dramatic boom and bust cycles. It lives in a coastal salt marsh habitat, and the populations would swing from, in a boom year, 14,000 individuals, and then a bust year would have fewer than 100 plants. So very concerning, but with the multiple decades of long-term monitoring, they discovered that these boom and bust population trends were explained by changes in rainfall, temperature, salinity, and even tides that were associated with an 18-year lunar event cycle. So a lot of factors going into explaining its population trends, but it took that long-term monitoring to figure it out. Okay, and regular monitoring will also lead to just a better stewardship of the whole outplanted site. It may need ongoing management to provide the best habitat conditions. So things like prescribed fire, invasive plant removal, and canopy thinning might be needed, as well as fencing or caging to protect the plants from herbivores. But there are many complications with these types of long-term projects, and continued funding, again, being one complication. Also, the issue of staff turnover being another complication. These are the types of things that can derail an otherwise solid project, so they just need to be planned for. And finally, we include disseminating results, reporting on results as a very important project component, because there is a strong bias in scientific literature to not publish negative experimental results. But we know that it really takes the honest, full, complete, honest reporting. It's such an important part of the scientific method, and it's especially worthwhile in these newer and developing fields like rare plant reintroduction and recovery. The detailed descriptions of methods and results and successes and failures and unexpected events will all help others to learn and advance the science. But as long as a project is done well, any outcome can provide useful ecological information. So that was just a brief summary of the topics that a reintroduction project needs to take into consideration. There are many more details in the report. And also in our report, we include a checklist for those who may want to design a reintroduction project. And all the credit for this checklist goes to the Center for Plant Conservation. This is a checklist that they created. I just kind of condensed and reworded it. But it's written as a series of questions and considerations to address at each step in a reintroduction project. So starting with the early stages of research about the biology and ecology of the target species, to the funding, legal, and regulatory considerations to ensure the project has a sound foundation. And it then moves on to the stages of implementing a reintroduction project. And it asks questions related to the experimental design. Where will the seed be sourced from and how will it be propagated? How and why were the recipient sites chosen? And what does the outplanting process look like? And there are questions about, are there plans for site management and monitoring protocols? And finally, reporting on the outcomes of a reintroduction project. And so just to give you a brief example, the section on sourcing propagules has questions such as, why are you collecting from a particular location? Have you thought through the when and the how, how many propagules will be collected? Will seed collection benefit the genetics for the founding population? And how will the propagules be stored and handled? So using this checklist as a guide when planning a reintroduction project should help folks to think more broadly and just to consider other aspects that they might not have thought of. And we end our report with a section on resources for folks to consult for more information and guidance. That includes other organizations, websites, all of my references, and a glossary of terms. Okay, so, I've said a few times through this presentation that reintroductions often fail, and I just wanted to quickly provide some of the statistics on that. In the report, I review and summarize 4 meta-analyses of reintroduction projects, but this here is just one example. So in 2011, Godefroy et al. Reviewed 20 years of reintroduction projects in the literature, and they also surveyed practitioners. And so it was a review of 94 species introductions. And most of them only had about 4 years of monitoring data that they reported on. And 52% of the projects reported survival of the outplanting, but again, that's just within the first 4 years. Only 19% of projects saw some flowering, and about 16% of projects saw some fruiting or seed set of the reintroduced plants. So it seems that the longer that a reintroduction project is monitored, they tend to see more mortality. So success rates decline. And this last point speaks to the bias in the literature, where survival rates that are reported in the literature are much greater than when practitioners were surveyed about their experiences with these types of projects. And also in the report, I describe a few examples of these types of more risky reintroduction projects. I just want to say maybe they're a little bit more risky. And so some of those features of a kind of a higher risk reintroduction project would include things like, for example, removing the entire adult plants from the natural population to transplant. That seems to be very risky and likely to be unsuccessful. Also, planting outside of the known historical range, otherwise known as assisted migration. Not that it should never be done, it's just a bit riskier. Not using an experimental design to properly test hypotheses. Using any intense site disturbance in the project would be a risky methodology. Not having full understanding of your target species as well is just, these are the elements that kind of lend to a just riskier project design. So maybe should be avoided. Sometimes, each case is individual, but should be thought about cautiously. Okay, so I just want to close by reiterating that rare plant reintroductions can be a great tool for conservation, but it's just not the right tool for all cases. But if it is justified, then it has to be done with the utmost caution at all steps in the process, and with a full understanding that you're undertaking a very long-term commitment. And so our goal here is to provide this document to be a resource for practitioners and managers and decision makers to just have a better understanding of reintroduction science, so they can have the information they would need to make decisions on these types of projects. And so I want to thank my boss, Bob Kartika, and these other folks with the New Jersey Department of Environmental Protection for all of their assistance and input in writing this report. So thank you so much for listening, and I'm happy to take any questions.
So the first question is, can you expound on the term genetic depression?
Oh, okay. I'm not a geneticist, but my understanding is that if you have a species population that's been in decline for any reason, but imagine the population has been in decline, and so you may have had 1 million individuals at some point, and maybe now you're down to 100 individuals. You can logically assume that the original genetic variation of the species has been reduced. So that's genetic depression, I think,
Okay.
in a very simplistic way of explaining it.
Okay, so do you have any information on the use of the growing mediums affecting the soil ecology of the site where the planting happens?
No, I didn't get into those details about the mediums in my report.
Okay.
That wasn't something I went into detail about. Although I will say, I think this is partially answering your question. A lot of, at least one of the meta-analyses showed that when plants were bare root planted, there was higher survival versus if they used the potting soil to outplant. So that's just one result that kind of pops in my mind related to your question.
Okay. So in cases where remnant populations of rare plants are growing in a threatened habitat that has different characteristics than described in the literature, well, how does one take that into account with planning and outplanting reintroduction protocol?
I'm not sure if I caught all of that. Can you repeat the question?
In cases where remnant populations of rare plants are growing in a threatened habitat, which has different characteristics than described in the literature, how does one take that into account with planning an outplanting or reintroduction protocol?
Yeah, I mean, I imagine you would need to think about why that would be the case. I mean, maybe it just hasn't been documented that that species can tolerate those different habitat conditions, or potentially, and this was discussed in the literature reviews that I read, are you looking at a site that's been degraded, and so maybe it's hanging on, but it's not in its preferred habitat conditions if it has been degraded too much. So that's a possibility. It's a complication, certainly. That's a tough one, but yeah, it's not something to be ignored, and maybe it is a case of degradation. If it's not, then that's fascinating. Then you have a case where your rare species is tolerant of different conditions than had been documented before. And then you're kind of doing original research and forging the way.
So have there been projects to increase rare species within its current microhabitat? And then maybe with more individuals, other seed dispersers could help with that new plant population.
Okay, can you repeat that again?
Yeah, have there been projects to increase a rare species within its current microhabitat?
Mm.
And then if there are more individuals, do other seed dispersers help with the new plant creation? So I guess if you're reintroducing to their current habitat where I guess they were found.
Yeah, I think that's referred to as an augmentation, and definitely Tennessee coneflower is one example where they found outplanting sites that some were unique and some were within remnant populations. And then for seed dispersers.
Well, I think what the question was, if there was, would that help with would they be around their seed dispersers? Would that help?
Oh yeah, yeah, that makes a lot of sense. Yeah, I think an ideal part of reintroductions is hopefully their original habitat microsites can be, are not so threatened and degraded that they can be repopulated, and then they'll have, they're all their associated species below ground and above ground, and yeah.
Okay. So we hear a lot about the risks of genetic outbreeding and depression. Has anyone looked into the risks associated with genetic fragmentation in the current era? And the other part of that question is, how can one determine when genetic rescue is appropriate versus a risk factor?
Yeah, a lot of the resources I read spoke to problems with fragmentation. This is outside of my realm. Like I said, I'm not a geneticist, but from what I've read, the procedures are so much more widely available, and they're less expensive than they ever have been. And so it's not as what's the word, out of reach to do genetic testing. If you can link up with the right people, it's less expensive and less complicated than it has been in the past. And so it is an important component, and should definitely, if you can, pursue that to be sure you're on the right track with the reintroduction.
Okay. And this will probably be our last question. Based on reported success rate, it appears the concept of moving plants from a proposed development site as a method for mitigation may be a red herring that agencies should avoid approving. Do you agree with that or have an opinion?
My opinion is to agree with that statement, that it's probably not in the best interest of the species and should be looked at very critically.
Liz, I'm going to chime in on this. I'm Bob Kartik. I'm administrator of the Office of Natural Lands Management. I'm also very proud to say I hired Liz Olson. So this gets into a really critical question as to why we were interested in conducting this research and creating this report. As a government agency, we've observed that, some consider this, the propagation of plants and use of propagation as a conservation technique to be kind of a no-brainer. A species and its future existence is in jeopardy. So why not grow it from seed and plant it all over the place? Or, as the example that you provided here, there's this housing development is proposed. It's going to take out a rare plant species. Well, it's a plant. We'll just move the plant. Problem solved. And when I say people feel this way, a lot of people feel. I'm not just talking about the developer and their consultants. I'm talking about the project managers within the department who get these applications, and they say, "Sure, that makes sense. We want to make these projects work. We want to conserve the species. Here we can accomplish both goals." Well, maybe not so simple. Maybe you should be thinking about some other things rather than just moving the plant. Do you want to? Does our department want to create this precedent where we are kind of paving the way to use to turn to plant propagation and moving adults and moving and trying to establish seed in other locations? Should that really be mitigation for a project? I've argued no, it isn't mitigation. We shouldn't be using this as a conservation technique in cases like that, we should be looking at other things. So this report is really intended to just give people, including people with my own department, a lot to think about before they make these decisions, which could be critical and could set precedent for future actions that, you know, might come back to haunt us. So it's a good question, and it really gets to how we feel this report should be used. You know, there's a lot of questions you need to ask yourself before you do what seems like the obvious thing to do, you know, that most people would agree to do. So thanks for the question.
No, thanks, Bob. Well, we are now a little over 1 p.m. I just want to thank Liz. Thank you for taking the time to present with us today. Bob, for jumping in and sharing this information with us. And I did post links to the plant report that Liz referred to, as well as to the report that New Jersey did on rare plant propagation and reintroduction. So those resources are there for you. If your question did not get answered, I will be sending an email with a link to the recording and Liz's contact information. And I'm sure if you still want to ask the question, she'd be happy to respond to it for you. So thank you very much. Thank you for joining us today. And we look forward to seeing all of you again in 2022. Thank you.
Thank you.